The purpose of this project is to test the development of 3D printed biorenewable polymer aqueous tissue for tissue engineering. So what is tissue engineering? Tissue engineering is the biomedical engineering discipline that uses the combination of biology, chemistry, physics, and engineering to test for suitable biochemical and physiochemical factors to restore, maintain, improve, and replace different types of biological tissue. There are three different sectors: improvement, development, or restoration, and this project focuses on the development of tissue-mimicking hydrogel scaffolds as a solution for transplantation and chronic disease. So this was done using hydrogels, which are also known as biorenewable polymer aqueous solutions. They are a 3D structure using interweaving patterned pores that can host chemicals that propagate life. In this case, can grow the cells that basically form it into a living tissue. The concept of this is implantation. So the cells, once isolated from the patient of the diseased area, are grown in an expansion and then injected into the scaffold, which holds special growth factor chemicals that allow that certain type of cell to grow and thrive, allowing it to form basically a secondary tissue. So this was done using 3D printing, which after forming a physical 3D model using CAD was developed into two types of shapes, a cube and a grid. And the reason why this was done with 3D printing was because most 3D printing plastics are environmentally toxic because they're petroleum based. So the focus of this experiment was to develop a renewable and biodegradable resource as well as create a hydroprinted 3D scaffold. So, as said, the goal of this project is to develop a renewable polymer solution that compared biochemical characteristics of three ingredients of hydrogels. First, cellulose nanocrystals, or CNC, type A gelatin, or porcine gelatin, and type B gelatin, or bovine gelatin, when combined with sodium alginate to develop a renewably sourced 3D printed scaffold. So, this project was done by creating three separate solutions, the type A, type B, and CNC, and then exposed the solutions to rheological and mechanical testing to see if they were even suitable to go through the 3D printer, which once the 3D printer would accept or deny them, would print the pattern scaffold, and then they would be introduced to a simulated biological factor or biological agent and chemical. So, as you can see on the very far left, it is the type A of the first run, which proved that the type A was going to be very runny and very viscous, meaning that when compared to three solutions, the most firm turned out to be type C and C, as can be seen in the middle of the second picture to the left. Once it went through the frequency testing of the rheological test, we were able to see that on the very far right, the very last picture on the right, it formed a sort of shape that was able to elastically hold or malform according to the frequency it was exposed to. The 3D prints on the very far left, those two pictures show the grid structure that is being printed, and it was done to mimic how the actual scaffold would look. Meanwhile, the third picture, or the second to the right, showed a CNC grid, which basically simulated what the grid would look like up close, since there are nanopores in the ones to the left. And then, as you can see on the very far right, it's an image of the printing the type B cube as necessary, using frozen plates in order to solidify hardening quicker. So, as you can see in these three pictures, there are very different curves shown, but the one in the center is the one to focus on because the type B is the only one with a positive slope, which is why it's the most suitable to the standard solution that is being compared to. Once it has a positive solution, it means that it is very flexible, and it also has a very adjustable and adaptable viscous solution that was most wanted for this experiment. And then in this picture, I would like you to also focus again on the center graph, which is the type B moduli. It shows that the type B is very elastic and very accommodating with a high tensile strength and high heat capacity, fitting the standard of a normal thread. In conclusion, type B was proven to be the most viable because of rheological and printing and solidifying as quickest of all of them. The CNC did prove to be too thick of a base compound, and it needed some refinement to work with other structures, and the grid matrices did have to be adjusted between solutions to account for spread, like in type A,